Hello student, today we will discuss some aspects which are important from examination point of view, and this thing is just you have to cram, you have to memorize them again and again. So human leukocyte antigen, which are also called major histocompatibility complexes, which are encoded by chromosome 6, and there are two type 1 and type 2. Type 1 they include A, B and C, and type 2 include D, P, DQ and DR. Now, HLA-A3 is associated with hemochromatosis, and B51 is associated with Behçet disease. B27 is associated with ankylosing spondylitis, psoriatic arthritis, and reactive arthritis, while DQ2 to DQ8 is associated with celiac disease. DR2 is associated with narcolepsy and Goodpass. DR3 is associated with DPS, that is dermatitis herpetiformis, primary biliary cirrhosis, and Sjogren's syndrome. And DR4 is associated with type 1 diabetes mellitus and rheumatoid arthritis. Then some problems, they are associated with deficiencies of some components of complement. For example, if a person is deficient in C1INH component, he or she will have hereditary angioedema. Then C1Q and C1R and C2 and C4 deficiencies, they lead to SLE and Knox-Schonlein purpura. C3 deficiency, it leads to recurrent bacterial infections, and C5 deficiency, it leads to is Liner's disease. It's wrongly spelled here. It is associated with dermatitis and diarrhea. And then C5 to C9 deficiency predisposes an individual to meningococcemia. Then after that, another important exam question is to know the functions of different organelles in the cells. First of all, we'll discuss about endoplasmic reticulum, which is of two type: rough endoplasmic reticulum. It is called rough because it has got ribosomes which manufacture proteins, and smooth endoplasmic reticulum which form different hormones which are lipid in nature. Then is the Golgi apparatus, where packing of already formed molecules takes place, and they are tagged with mannose 6-phosphate and are directed to lysosomes. After that, we will discuss about mitochondria. Mitochondria is the place where aerobic respiration takes place. It means that food is broken down in presence of oxygen and it generates energy. Mitochondria, they contain circular DNA. Then lysosome, where breakdown of proteins and polysaccharides take place. And after lysosome, we will discuss about peroxisome and proteasome. So peroxisome is actually related to the breakdown of long chain fatty acids, and proteasome is associated with degradation of protein which is tagged with ubiquitin. Then after that, nucleus, which contains DNA, and from DNA messenger RNA is formed, and plus splicing of RNA also takes place. It means that unwanted and unnecessary RNA or sequences, they are removed. Nucleolus, it forms ribosomes, and ribosomes in turn, they form proteins. So this was all about cell organelles, and after that I'll show you a picture where you can see an animal cell, and in this animal cell you can see nucleus, and you can see smooth and rough endoplasmic reticulum, lysosomes and mitochondria. Then after that, there is another topic which is very, very important from examination point of view. It is Turner syndrome, and its karyotype is 45X0, and it's a short stature female with webbed neck, and patient has got widely spaced nipples, short fourth and fifth metacarpal bones, and then abnormalities in the heart like bicuspid aortic valve or coarctation of aorta. Then there can be pigmented nevi over the body, and cystic hygroma, lymphedema, and there can be horseshoe shaped kidney, and these are the patients of primary hypogonadism, means that their ovaries, they do not work properly, so they have primary amenorrhea.